So we're finishing off, and this in a way is a discussion of the nervous system, and really the peripheral part of the nervous system where we're detecting stimuli and bringing in information about our external environment. And that's what we're mostly talking about, external environment. But we're going to also talk just a touch about internal sensations that feed into the central nervous system to let us know the status of our body. And so we have a number of senses that we're going to talk about today. We have 5 special and several that are called somatic. Sometimes they're called general senses because they're distributed throughout the body. But whether we're talking about specialized or general, all sensations are going to involve the stimuli that is able to then cause a reaction from a receptor. And our receptors are going to be proteins that are able to detect the stimuli. And that detection of the stimuli is going to basically set off some biochemical reactions that will result in an action potential. And then we get into the nervous system and the relaying of that action potential to the brain. So receptors really are transducers. They take mechanical stimuli or chemical stimuli or light stimulating a receptor. And they transduce that stimuli into an electrical signal that then feeds into our nervous system. So let's look at a couple of these stimuli. We can categorize some as mechanoreceptors. Now, these are those proteins that are going to be detecting some type of movement, mechanical energy. Hearing. What is it we're detecting when we hear sound? Sound waves. These waves of pressurized air moving through the air, impacting on our eardrum. We're going to talk about the components, anatomically speaking, of our organ in which we can detect these sound waves. Balance. Now, balance is going to be another one of our specialized senses. It's also located in our inner ear. But with balance, we have some interesting structures. And the arrangement of these structures and hairs such that we're going to take advantage of the law of inertia. And if you're not familiar with the law of inertia, maybe you can Google it before we get there, but we're going to talk about the law of inertia and how your body uses it to maintain balance, but also to give your body information about, are you standing still, are you accelerating, are you decelerating? So this is almost like a car. But that's going to be balanced in our discussion, but it's very mechanical. And touch, which is now considered a general sense. It's no longer a special sense, because balance is simply located in your inner ear. Where can you feel touch? Everywhere. So it's a somatic or a generalized sense, but that's pressure. Again, a mechanical sensation. Thermoreceptors. Okay, so who this morning walked out of your dorm, of your apartment, of your house, and it was cold? Yeah, we detected that really fast. So do we have cold receptors in our body? That's a trick question. Do we have cold receptors in our body? No. We have heat receptors. And what is cold? The lack or the absence of heat. It's just like we have light detectors, right? Do we have dark detectors? No. What's dark? The absence of light. So we have these receptors, and if there's a lot of heat, it's going to register. If there's not a lot of heat, it's going to register, and our brains are going to say, wow, it's cold. Let me go get a jacket. But those are our thermoreceptors. We have pain receptors. Now, pain could be extremes of pressure. Have you ever had those individuals shake your hand and you feel like they're trying to break your knuckles into powder? just because. So that's pressure. That's a painful situation. We actually have, in some of our research labs, liquid nitrogen. With liquid nitrogen, you have to use very heavily insulated gloves because it is cold. And if you happen to spill liquid nitrogen on your skin, or maybe you've done this with dry ice, maybe at a party or at Halloween when you're trying to make the witch's cauldron. You ever played with dry ice? Have you ever touched dry ice for any period of time? Does it feel cold? What does it feel like? It feels like you're burning your skin. Because the same way that excessive heat damages your cells, excessive cold damages the cells, and it's painful. So extreme cold is going to feel the same as extreme heat. And it's going to cause damage, and it's going to activate the pain receptors to tell you, stop doing whatever you're doing. Or fix whatever's wrong. That's what pain is. It's a signal to fix something. Now, our chemoreceptors, receptors that detect chemical sensations, these are going to be sort of a tandem pair. Because we have taste, which are going to be the taste buds of our tongue, but also in our olfactory spaces, our nose, our sense of smell. And they're actually going to work together because when you have a cold, does food taste the same? Does it taste kind of bland? Because the chemicals that also get into your nasal passages enhance the flavor and your enjoyment of the different flavors that you're actually tasting on your tongue. So they're going to work together. But these are all of our chemical sensations. And then finally we get to our photoreceptors, meaning light detectors. And these are our specialized receptors that are in the back of our eye, in our retina. And we used to say some detected black and white and some detected color. Well, now they're saying some are active in low light and some are active in bright light, which is kind of saying the same thing. Low light, black and white, or grayscale, whereas bright light, vivid colors. And we're going to talk about those two distinct types of photoreceptors present in our retina. So as I mentioned earlier, here are our 5 special senses. And you could probably get these if you think about it, with balance being the one that I would highlight. If I'm you, I'm going to highlight balance because that's the new one. That's the tricky one because most people say smell, taste, sight, hearing, and touch. But we had to demote touch, and we promoted balance to a specialized sense. Or, as you see here, a somatic or a generalized sense. These are specialized because they're located in one place in the body, whereas our somatic sensations are covered over our entire body. So we have touch receptors over our entire body. What other receptors do we have over our entire body? Touch. What else can you detect? On your fingers, on your toes, on your legs, on your arms? Pain. You got a cut on your leg, it hurts. You got a cut on your arm, it hurts, right? Pain. So we can detect touch. We can detect pain. What else? Heat. So temperature. So those would be among the few of our somatic or generalized sensations for our body. So let's start with taste. When we look at the surface of the tongue, we are going to see a number of little spots on the tongue. These little spots are called papillae. And on the sides of some of these papillae, especially on the edge of your tongue and on the back, there's a little V in the back where we'll see this in an illustration coming up. We have a very high concentration of these specialized cells that are called taste buds. They're put together into a little barrel-shaped structure. We have some cells that are supporting cells that help maintain the shape, help nourish, help protect. And then we have our actual taste cells that we see here highlighted in green. And then we basically have these dendritic types of sensory nerve cells that are going to be activated to form an action potential when these nerve cells actually detect chemicals. And these chemicals are called tastants. Now, if you look at the surface of the epithelium of the tongue, the epithelium is in pink, there's a little pore. It's called the taste pore. I know it's a real original name, right? But extending just out of the taste pore are these little processes from our receptor cells, and they're called taste hairs. And it's going to be on the surface of those little cytoplasmic projections where we have the actual protein receptors for certain tastants. So the chemicals bind to the receptors on the taste cells. That causes a change in the ions inside the entire taste cell. And that voltage change is going to be registered by the neuron to reach the threshold for an action potential. Boom. Then we send a signal, goes to our brain. Oh, we tasted this. And it tastes like whatever it is you're eating. So that's how the taste gets started. So here in our illustration, you can see the edge of the tongue, a very high concentration of our taste buds, and here's our little V-shaped structure near the back part of the tongue where we have another extremely high concentration. You can actually find taste buds scattered all over the surface of the tongue, but this is where they're going to be most abundant, in those shaded areas. Now, if you printed out the notes or if you printed out the PowerPoint slide, I have added an additional taste. It is now an official taste, so instead of 4 basic tastes, there are 5. And some people are trying to add 6 really weird ones, and we're not even going to talk about those. But when we talk about taste, we talk about sweet, sour, salty, and bitter. And now the 5th taste that really is very commonly accepted nowadays is called umami, which is a savory kind of taste, savory kind of flavor. And it was thought that you had specialized taste buds that only detected one or the other. It used to be that your tip of the tongue was thought to be the sweet place and bitter was on the side. No, most taste buds are versatile. Some are going to detect some more than others, but they really can detect almost every one, and that's going to happen all over the rim of the tongue as well as this terminal sulcus there in the back. So sweet, sour, salty, bitter, umami. Some of the new ones are talking about calcium, and some of these others I'm like, okay, we're just going to stop with these normal ones and then throw in umami, which I guess is a sort of an Asian kind of culture flavor that is very commonly accepted now. Now, we can't talk about taste unless we talk about smell. And although we have 5 sort of basic tastes that we have, there are thousands of different odorant chemicals. So we're not going to try to list all the different smells and categorize them into just a few categories, right? Because you'd have to have sweet, you'd have to put in BO, you'd have to put in funky. I mean, you know, there would just be way too many. But with all of these odorants, tastants were for taste, odorants are for our smell. These chemicals are going to get brought into our nasal passages. And inside your nasal cavity, you're going to have these ridges of tissue that have these receptor cells, and these ridges increase the surface area so you can pack more receptors in there. More cells, more receptors, a higher sort of fidelity of being able to smell something. And if we look at the very surface, the very top portion or superior portion of our nasal cavity, this is where we're going to have a really high concentration of our receptor cells, and they're adjacent to the beginning of these nervous system cells that are going to relay this information to the brain. So our cells are referred to as olfactory cells or olfactory receptors. Olfaction means smell. And so in some ways, these are going to look similar to our taste cells in the sense we're going to have these little hairs that are going to stick up and these chemicals are going to bind to them. It's going to cause the ions to change in the cell, and that's going to signal the nervous cell. So it's going to be fairly similar to what we've seen in our taste buds. All right, so our olfactory cells have these olfactory hairs. And once we bind the chemicals to those olfactory hairs, we're going to signal down to our nervous system cells that then get to the brain and say, okay, there's an odor. And your brain's going to decide, is it pleasant? Is it noxious? Do we not really care? It doesn't smell that bad, doesn't smell that good. But this is the thing that blows my mind. We had 5 basic tastes, right? We had 1,000 or so chemicals. When you combine the sense of taste with the sense of smell, you can, in fact, distinguish and discriminate over 10,000 different sensations. Because taste and smell are going to complement and work together with each other. I do not have that capacity. You've seen blind people, right? That's fairly common. And you've interacted with deaf people. That's pretty common, right? I have no sense of smell. None at all. And people usually look at me like I'm a freak of nature. Oh, you can't smell? Really? The very first question I get is, well, can you taste anything? I'm like, yeah. And there's some things I like, and there's some things. And just to give you an example, when it comes to steaks, okay, who likes just a perfectly cooked steak? Anybody going to admit to you a meat eater? You like a perfectly cooked steak. Okay. Does a T-bone steak at a great restaurant, does it taste different than a hamburger patty you get at McDonald's? Not to me. It tastes exactly the same. So I don't care how much. I know. I don't care how much you spent making that steak, how much you paid for that steak, what 5-star chef made that steak. What am I going to ask for to put on my steak? Ketchup. And people look at me like I should be arrested for putting ketchup on a steak. You put ketchup on your steak? Oh yeah, sister girl. Oh yeah. But it's the sweet. When you think of ketchup, it's really sweet, but it's got the acidity too. It has to be really strong for me to detect those flavors. Now, I don't like hot, spicy stuff, but I do like spicy food. Probably more so than most people would. Yes, ma'am.
Question.
Okay.
And this is kind of about my nose. So I say I have no sense of smell sometimes, but I do. But some things I just don't smell. Like I'll be in like a hospital and like someone in a diaper and they're like, oh, it smells terrible. And I'm like, what are you talking about? Like until I get really close, can I smell it?
Okay. So there may be your smell's certainly not as heightened as some people. Some people have super sniffers, and it's usually moms. I don't know. Mom can always tell where you've been even before you get in the door, right? You walk in the door, and Mom's in the back of the house, and Mom's like, "Why'd you go there? Why'd you go over to so and so? I smell it." I don't know. My first wife. Let's get back to the story of the diapers. My first wife, with our first child, she had a super sniffer. And so when the baby messed up the diaper, she would smell it. And she would go get busy doing something and say, hey, honey, could you go check to see if the baby's diaper needs to be changed? I am such an observant scientist, it took me 3 months to figure out what was going on. She was sitting reading the paper one day, and she smelled it, and she went, phew. She put the paper down really fast. I'm like, what's going on? And she got up and went straight to the kitchen and started washing the dishes. I'm like, oh, that ain't right. Something's wrong. 5 minutes later, she said, would you check the diaper? I'm like, and I went in there, and it was, okay, I graded diapers on how many wipes it took. Okay, this one was a pen wiper. If you've changed the diaper, you feel my pain, right? And I knew. I was like, dadgummit, she's smelling it. And she just lit me up. So the very next weekend I had off, I followed that baby around for about 2 hours, checking that diaper every 2 minutes until she loaded it up. And what did I do? I went and got busy. Hey, baby, could you check that diaper? Sure. Because I can't smell, right? About 10 minutes later, she comes walking back into the kitchen with that diaper to put in the pail. She said, what, did you follow her around all morning? No. I would never do anything like that. But I couldn't smell it. And you've seen some of those videos of the guys trying to change the diapers and just about losing their lunch over it. So it's not all bad that I can't smell. There's a lot of good smells you're missing out on too. Here is a really close-up illustration. Down here, this is the odorants, the chemicals, and that's going to be dissolved in the liquid in the mucus that's in your nasal cavity. So mucus isn't always a bad thing. And when those chemicals bind to those little hairs that you see sticking out into the surface, once again, binds to the receptor, changes the ion concentration inside the olfactory receptor cell, that's then going to signal a neuron, in this case our olfactory neuron, that sends the information to the brain. Oh, we smelled something. Oh, it smelled like this. The more chemicals that bind, the stronger the smell. The fewer chemicals that bind, the weaker the smell. All right, so taste, smell, now hearing. We're pretty familiar with the fact that hearing occurs in your ear, right? So we can localize it there. But when we say the ear, understand there are 3 parts. We have the outer part of the ear, which is basically the external ear. This flappy thing out here, some are big, some are small. Some earlobes are attached, some are not. And we call that external part the pinna of the ear. Then we have the external ear canal, that thing that you should never put a Q-tip in, that I just cleaned out with a Q-tip this morning. Right? That's a dad. Do as I say, not as I do. Right? So that's the external ear. The external ear ends at the dead end of your auditory canal at the eardrum. The big fancy name, tympanic membrane. And so those sound waves are funneled into your auditory canal by the pinna. It directs the sound waves down to that point. The sound waves travel down that tunnel and hit the tympanic membrane, and much like the head of a drum vibrates to make sound, the tympanic membrane vibrates. So it transports those waves that are moving through the air into the mechanical wave, and that energy is transferred to the eardrum. So here we see the pinna in our illustration, the canal, and there is just a cut section through the eardrum. It's an oval-shaped membrane, but they've cut some pieces away, and that's going to divide the outer ear that you see listed up here at the top bracket from the middle ear, which is in this little compartment right here. This is where we have those 3 little bones, and that's going to be part of our middle ear. Yes, ma'am. You're still going to get sound waves coming in. You may not hear as well.
Because I cut half my ear off.
Oh.
When I was a kid, all of this.
Yeah.
And so, but I could still hear it.
Did you notice any change?
I mean, I was in the 2nd grade. I mean, it was kind of more muffled, but
yeah.
I could still hear
Now, and 2, and you may have done this experiment. Have you ever seen the tuning fork that you tap and it vibrates and it makes that hum kind of sound? Have you ever seen anyone do that and then put it on the bone, like almost the occipital bone right here in the back? You can't hear it like you normally do, but when they put it on that bone, that causes the vibration of those 3 little bones, and you hear it as if it's going in the auditory canal. It is really freaky. It's like, I'm hearing it, but I don't know how I'm hearing it. It's just the mechanical waves getting in a different way. So you'll still get some, and even vibration impacting on the side of your face can cause you to hear some sound, but not as well. So we've talked about the outer ear. Now let's talk about this compartment, the middle ear. And I want to point out in our illustration, and we'll see it again, that this middle ear is also going to have a connection to your oral cavity and the pharynx via this little tube that you see running down at the bottom. That is going to be our eustachian tube. All right, so keep that connection right there. Keep that in mind as we look at the middle ear. Now, both the outer ear, the auditory canal, and the middle ear, this is air-filled. These are air chambers because we need the sound to be able to vibrate through air. And it's because of the eustachian tube that is going to ensure that the air pressure in the middle ear is the same as the air pressure in our auditory canal. Remember we talked about that a little bit with our ears popping when you get in an airplane? So that was the eustachian tube that equalizes that pressure. And we need the pressure to be equal so the eardrum can vibrate effectively. And so the eardrum starts the process of detecting the sound waves. And then we have these three small bones. The hammer, the anvil, and the stirrup. If you just want to remember those names, that's fine. If you want to remember the big names, the malleus, the incus, and the stapes, you can do that as well. Those three bones are going to transfer the vibration from the eardrum to the actual structure where sound is going to be detected. But it's not just the transfer. You're actually amplifying, because of their shape and their position, you're amplifying the vibration. That's going to be further amplified in the inner ear when we get to the structure called the cochlea. So can you imagine vibrating the eardrum? And now you're vibrating the three little bones that are then finally, via the stapes, that's going to be connected to this thing that looks like a snail. And that's going to be the cochlea, and that's where we're going to have the receptors that detect the sound vibration, and that's where we hear a tone, or that's how we're going to hear a sound because of the cochlea. So where do we first pick up the sound waves? Eardrum. Transferred to the bones of the middle ear. That's then transferred to the cochlea in the inner ear. And those sound waves are going to be detected by over 15,000 mechanoreceptors. And these are going to be in just what are termed hair cells. Those bones are going to vibrate a little membrane on one end of the cochlea. And that's going to cause changing of the air pressure inside the cochlea. And that air pressure, like an air pressure wave, is going to run all the way down the cochlea and come back on the opposite side, almost like a racetrack. And as that pressurized sound waves moves, there are going to be these little hairs that are sticking up, and that pressurized sound wave is going to bend those hairs. And as those hairs get bent, and the extent to which they get bent, and the frequency by which they're moving and vibrating, that is going to determine what sound you hear. That's why if you are at a concert, and you're at a concert close to the big sound system, close to the speakers, and it's really, really loud. Have we all been there? When you leave that concert, have you ever left and your ears felt stopped up? You have so bent those hair cells that your body has accommodated. Meaning your body says, that's so loud, we're going to turn down the volume. It's like we're going to see with the visual system. Have you ever come in on a summer day from playing outside and everything in the house is so dark? There was so much light stimulating your retina that your retina says, okay, we're going to tune this thing down so that we don't hurt ourselves. That's what's happening in your ear. And if you continually expose those hair cells to severe bending because of loud sounds and noises, those hair cells are going to bend, and they're not going to bend all the way back. And you're going to start losing your hearing. So protect your hearing at all costs. My dad, he has never worn earplugs or anything with all the power tools and everything he's used all of his life. He's 80 years old. He is deaf as a hammer. I mean, he cannot hear. You should hear me try to talk to him on the phone. Dad, get your hearing aid. Get off the cell phone. You know you can't hear on the cell phone. So even if you don't think, oh, this is not very loud, I'm not going to take the time that it, no, put your hearing protection on, because once you damage it, you may not get it all back. And it only takes one time. One time. I told you my wife sings, and her last gig that she had at the Horseshoe Casino in Shreveport, the second night she did, I mean, the place was packed. So the guy cranked the amp, and her in-ear monitors weren't working that well. So they were like screeching. It took her a week and a half to get all of her hearing back in one ear, and she was panicked. Can you imagine having to sing and you can't hear out of one ear? So she was panicked. But it finally came back. But we're going to try to make sure that doesn't happen again. Does somebody have a question? I thought, okay. This is basically that snail-shaped cochlea when it's all stretched out. But there's a little oval window right here against the stapes. And when the stapes vibrates, because the eardrum vibrated, that's going to cause these sound waves to vibrate. Do you see how they run around like a track? And they're actually going to sort of be ejected or exit out of this, what's called the round window. But when you vibrate that air, your hair cells, and you can see them right here, those hair cells are going to be bent. They're going to move. And remember, there are over 15,000 of them here in the cochlea, that as they bend, that's going to be what detects the sound that's coming in from the outside. Now, the bending of hairs, we're also going to continue to think about when we get into balance. Now, our balance is going to be that part of that snail-shaped organ in our inner ear. The cochlea was the round snail-shaped thing. We have these other parts. We have the main body part, and then we have these three little looping things that are called the semicircular canals. The semicircular canals and the vestibule, the main body of this structure, are collectively called the vestibular apparatus. Vestibular because the vestibule being a part of that. Do you understand what the term vestibule means? Not scientifically, just a vestibule. Have you ever heard that term before? Y'all don't go to church at all, do you? You bunch of heathens. I remember growing up, and we would come into the church. You come in the front door, and there's that main room where you usually have the old people shaking your hand and everything and giving you the bulletins and the programs and stuff. And from there are a number of different doors you can go into. That main room with the different doors, that's called the vestibule. All right? So the vestibule in this case is going to get you to either the semicircular canals or the cochlea. So it's a non-scientific name, but it's kind of named after sort of architecture in this room and the building. But the semicircular canals, they are going to be involved in rotational sensation. Your semicircular canals are basically positioned three-dimensionally, so you have an X, Y, and Z axis to them. So that gives you three-dimensional information about where your body is in space. Whereas the main body, the vestibule, this is the one that's going to determine, okay, are you not moving? Are you speeding up? Are you slowing down? And with that information, you know where your body is in space, and you know at what speed or no speed your body is actually moving. Now, unlike the cochlea that was air-filled, these structures, the semicircular canals and the vestibule, basically have a gel-like fluid. It's thicker. And we're going to have our mechano hair cells sticking up into this thick gelatin-like fluid. Now, this is where I want to talk about the law of inertia. Did anybody Google that, look it up? Does anybody know the law of inertia? You probably do and you don't realize it. An object at rest stays at rest. An object in motion stays in motion unless acted upon by an equally forceful something in the opposite direction, right? So in this case, with considering both of these, when you think about the gel that's in my vestibule, Right now, what is that gel doing? The gel in my vestibule, what's it doing? Nothing. It's static. It's staying still. The hair cells are sticking up. But the second I move forward, what does the gel do? No. It's going to move backwards. Why? Because it's going to stay in place until it hits the back of the vestibule. Right? An object at rest does what? Stays at rest. So that gel is staying in place. So if you think of my fist maybe as that gel, as I move my head forward, it's going to tend to stay in that same physical location like a GPS pinpoint. And so relative to my body, it's going to move backwards. And as it moves, those hair cells are going to bend. And when the hair cells bend backwards, what is it telling my body? I'm accelerating. Now, as I'm walking around, what's going to happen to the gel? What's happening to the gel right now? It's not moving, right? It's going at the same speed I am, so it's staying in place. What happens when I stop? It moves forward, bends the cells in the opposite direction, tells my body, oh, we stopped. So you see how the inertia part works in the bending of those hairs? That gives you the static acceleration, deceleration. With our semicircular canals, it's the same thing, but when those hairs bend relative to what's happening in the vestibule, that says we're either moving to the left, to the right, to the front, to the back, up, or down. That's our three-dimensional patterning with our 1, 2, 3 semicircular canals. And again, this is not a great illustration, but again, the hair cells sticking up. This is a little ridge of material. You can think of that almost like the gel that's being moved and bent depending on which direction you're moving your body or how fast or slow you're moving. All right, so let's get to arguably the most complicated sensory organ in the body. Now, the cochlea was pretty complicated, right? With the cochlea, the vestibule, the semicircular canals. The eyeball is, to me, is just an absolute miracle that how this thing functions. And as we look at the different parts of the eye, and we're not going to get into the absolute fine detail of it, that just kind of goes beyond what we need to do in this class. We're going to talk about the sclera of the eye. That's going to be the white of the eye. And if you were able to take an eyeball out, you would see that covers the entire outer surface of the eye, the sclera. Except for the very front part that's clear. And that front part of the eye is the cornea. Now the cornea is the one that sometimes in older people it starts to get cloudy. What do you call that when the cornea gets cloudy? And they have to have this kind of surgery. Cataracts. Cataract surgery. That happens to the cornea. The clear part isn't so clear, and so it obstructs vision. Doesn't happen to everyone as they get older, but it's one of those things that just sort of creeps up on you with age. Now, if we look deep to the cornea, if we look underneath the cornea, now we have this disc-shaped muscle. It's a muscle because it can change shape. And that's the color part of the eye. It's called the iris. And the iris is going to control the shape and the size of the what? The pupil. Now, is the pupil a black spot? Somebody tell me, what is the pupil made of? It's just a hole. The pupil is nothing. It's just the space inside the iris. Kind of reminds me of the old Bugs Bunny show, the Roadrunner and the Coyote. Y'all maybe, have y'all ever seen these before? Are y'all too young? You've seen, okay. Have you ever seen the episode where the coyote digs a hole and then the Roadrunner comes along and picks it up like it's just a colored dot? It used to freak me out. I was like, oh, is that what that is? Is it just a black dot that's inside your eye? No, it's a hole. And the iris controls how wide or dilated or how constricted the pupil is. So light is going to pass through the cornea. It's going to pass the iris, go through the hole that is the pupil, and light is going to pass through the lens. Now, the lens in your eye is not like the glass or the plexiglass that I have in my glasses. It's not even like the flexible stuff that you have in your contact lenses. The lens of your eyeball is made of collagen and elastic fibers, very similar to what you might have in a ligament and a tendon. It's just arranged so regularly that light freely passes through it. And that lens, that organic lens, can actually refract light. Now, do you understand what I mean by refracting light? It can bend it. It can focus it on a point. And where you're wanting to focus it is on the retina, on the back of the eye where the receptors are. But it's flexible. The lens can change shape. Did you know that? Did you know the lens can change shape in your eye? How do you know your lens changes shape? Okay, do this for me. Put your pens, pencils down. Okay, we're going to change the shape of our lens. And we're going to do it almost instantaneously. All right? So I want you to pick a point somewhere on a wall, somewhere kind of far away from you, whether it's the fire warning light or that silver panel on the wall or that yellow box up front. Pick a point on the wall and just look at it. Okay? Find that point and look at it. All right? Now, hold your finger up close to your face and then focus on your finger. Are you able to focus on your finger? Now, look at that point on the wall. Now look back at your finger. Do you see how the focus changes? It's almost like you can see it come into focus like you're using binoculars. But you can sort of see it change, right? Those are the muscles that are pulling on the lens to change its shape so that you can focus either closely or focus far away. And that sort of gives you the example of how the lens of your eye is flexible. And as it flexes, as it changes its shape, it's going to focus the image on your retina. And it's important. You want that image to be in the right place, to be in the right focus, to be the right brights, if you will, so that your photoreceptors can detect what it is you're trying to see. All right, so let's look at this all together. So we have the sclera all the way around our eyeball, except in the very front. That's where we have our cornea. There's our iris. They've cut it in half so we can kind of look through there. There's the pupil. The light's going to pass through the pupil. And it's kind of like they made the lens look out like an onion. See how they've got those concentric rings? These are just the concentric layers of collagen that are laid down. And here's the little muscle that attaches to the lens to either relax and let the lens be round or contract to stretch the lens out. This is called the ciliary muscle. Then we have this big, huge space that is inside the eyeball, and it's not air-filled. It's not quite the consistency of the meat of a grape. You know how that's not liquid, but it's not solid? Not quite that solid, but we're going to have this gelatin-like material in here that's called vitreous humor. And light's going to pass through the vitreous humor. It's going to impact on the back of our eye, and that's where we have the retina. That's where our photoreceptors are. Now, we do have a spot in our retina that's called the fovea, and that's going to be the spot where we have the highest concentration of our photoreceptors. That's where we're going to have the highest visual acuity there in the fovea. So here are our two types of receptors that are going to detect light. And this is where we're going to make a slight change. Rods, because of their elongated shape, and we have about 120 million of these rods. These are going to be the receptors that function in low light and therefore grayscale. Again, we used to say black and white, but it's low light and grayscale. I think you can imagine if you're trying to detect very small amount of light, you want to have a bunch of them, right? But our second set of receptors are going to be dominant in very bright light, and this is going to help us detect those vivid colors, and those are the cones. They're much shorter, and they look like a little traffic cone, whereas our rods are going to look like just a hair comb that you might see at the barbershop. So grayscale are the rods, and cones are going to be color. I like saying that because cones start with C, color starts with C. That's how you can remember that. Rods, that's the other one. And that's the grayscale, black and white, and low light. This conditions. Now this is bizarre to me. We're not going to name all the layers of the retina, so don't panic. But it would reason to me when I first started looking at the anatomy of the eye that if you had these receptors that were detecting light, especially those that were in low dim light, the grayscale, it seemed to me you would want those receptors right on the surface where the light would get to it, right? No, they're upside down in the retina. You have all of these layers of neuron cells. You have ganglion cells. You have bipolar cells. There's actually about 8 layers of nervous system cells in between where the light is and where the light's coming in and where the actual rods and cones are at the very back. It seems to me all those cells are going to kind of disperse the light. It's going to be harder for the light to get in. But if some research has shown that the arrangement of those nervous system cells actually helps refract the light even better. But regardless, the light comes through, and as it's passing through, it activates the receptors, the rods and the cones, just before the light impacts the back of the retina, and there's cells back there that are pigmented. And the amount of pigment these cells have and the type of pigment they have can actually enhance your visual acuity at night. So for us, our pigment layer doesn't reflect the light hardly at all. So we get one stimulation. A photon of light comes in, activates the receptors, hits the pigmented layer, and usually is dispersed. The energy is gone. But when you look at nocturnal animals, do we know what nocturnal animal, what that means? What are nocturnal animals? They are awake at night, they hunt at night. You know, whether we're talking about coyotes or deer are awake often at night, raccoons, they're always out at night. I like to, okay, there's some TV shows where it's just the same stuff week after week after week after week, but it's like a train wreck you can't not watch. One of those shows is Swamp People. You know what I'm talking about? It's the same stuff. Every week, I'm like, well, just shoot it. But I can't watch it. Deadliest Catch, that's another one. It's crap. Every week, same thing. Crap. Why am I watching this? Wicked Tuna. I don't know what it is. But have you ever seen those guys go out at night? How do they find the gators at night? They put the light on them and those eyes just light up. Do you know what that is? For those nocturnal animals, this pigmented layer is heavily pigmented, and that layer is called the tapetum lucidum. You don't have to write that down. But that layer reflects that photon of light. And so for those animals, they can see at least twice as good as we can at night because the light passes through, stimulates the receptor, hits that reflective layer, and comes back and stimulates the receptor a second time. So it amplifies the light, and when you're shining the light, it's hitting the tapetum lucidum, and it's coming back to you just like a reflector on a car or a bicycle. And it gives them twice as much stimulation for each photon of light that they have. So do you see why they call them rods? And you see why they call them cones? I mean, that's like a traffic cone. And they have all these membranous folds that have all of these different proteins on the surface. Rhodopsin is going to be one of these proteins. And rhodopsin is important. And guess what one of the vitamins that you take in your diet is important for developing rhodopsin? Vitamin A. What vegetable has a lot of vitamin A? Carrots. You ever heard that? Eat your carrots, good for your eyes. Well, it's one of the places that came from. Carrots aren't the highest in vitamin A, so I'm not really sure how that got put together. But carrots do have quite a bit of vitamin A. All right, anybody colorblind? Anybody colorblind in here? Exactly. And that's what I was looking around at the guys, seeing if any of the guys raised their hands. Because the gene for colorblindness is a recessive gene, meaning you've got to have the colorblindness gene from mom and you've got to have the colorblindness gene from dad. Ladies, you do if you're colorblind. Because that gene is on the X chromosome. And you've got 2 of them, right? How many X chromosomes do guys have? 1. That's why guys are more often colorblind than ladies. But guys, if you're colorblind, you can't blame dad. Who'd you get your colorblind gene from? Mom, because where did your X chromosome come from? How do you know it came from mom? You had to get the Y chromosome from dad, because mom doesn't have a Y chromosome. So the mother inherits the colorblind gene to the males. And it may be as high as 8%. Females, 1% or lower are colorblind. And the typical colorblind is the red-green colorblind. You can't distinguish those. All right? Now, no one said they were colorblind, right? Okay, we're gonna test. All right? Don't yell out your answers. All right? Play fair. Okay? Write down the number that you see on the screen. And yes, there is a number there. What number? Oh, you colorblind, dude. Does anyone not see a number? Anyone not see a number? All right, so we've got the number 21. No, I'm messing with you. 12. Oh, man, I am colorblind. I got you all. I got you all. I got you all. All right, number 12. Anyone not see 12? Okay, number 12. Next. Ooh, okay, let's start up here. That's the easy one. You know, if you can't see that one, you're not just colorblind. You're just blind, period, right? All right, that one's pretty good. Okay, what do we got for our number here? 25. Starts to get a little bit harder, doesn't it? All right, let's move over to the middle. What we got? 29. All right, let's give everybody a chance. Get your eyes adjusted. What's our number? 45. Okay, that's pretty easy. We got 56. Woo, I'm really having to look down here. Y'all got that quick. What about over here? 8. All right. Do we have trouble? Dude, colorblind? Which one's hard? Which one's difficult for you? Now, which one of our set here? Now that you see the numbers? So 6 and 8? Yeah. So you can barely see that one? I mean, these are tough. Those are tough, but you can't see those at all. Okay. Yeah, so there's a partial color blindness. If you go in the Army, just fake it. Okay. So that's a partial one. I think this one is supposed to be a hard one. I'm not sure. I can't remember. Yeah, they won't take you if you're colorblind.
Is that like a new rule?
I think it's been a rule for a while.
My grandfather was in the Army and he can only see gray.
He lied. He lied. Well, now, when was he in the Army?
He was in Vietnam, I think.
Oh, yeah, Vietnam. They would take anybody. No, you had a pulse. That's called drafted. No, you didn't have a choice. All right, I think we got one more, and I think this one is supposed to test a different kind of color blindness. Let's see what we've got here. Ah! Well, it looks better there. It's a creative brain thing. Okay, I'm seeing. Okay. See, this right here looks like it's almost. Okay. I've got a 2.
Yeah, I want to say this gave it away.
Yeah. I got a 2. The little tail, the little swoof right there. I got a 2. All right. I mean, anything? Okay. This is tough. I mean, this is tough. I tried to look for one that was like an optical illusion just to mess y'all up, but I couldn't find one fast enough. So, all right. Now, color blindness, that's one you either have or you don't. But ones you will encounter probably. I see a lot of people with glasses. We've got contact lenses you're wearing. So the shape of your eye, that can affect your vision. You can either have myopia, which is nearsighted, which is what I have. I can see just fine, like if I'm reading without my glasses. But right now, I wouldn't even know who you were back there. I mean, sir, ma'am, no. Yeah. I'm not quite that blind. But that's nearsighted, myopia. Hyperopia is farsightedness. You got that just fine, but you can't see up close. Now, most of us are going to end up with the hyperopia of old age, right? We all know what that looks like, right? It looks like this. This is what hyperopia of old age looks like. Right? Because you can't see up close anymore. You have, again, it's the changing of the eye, and it's different than what you normally see in your everyday world. That's why a lot of people that get that hyperopia of old age have what? Reading glasses is one. Bifocals. They have bifocal contacts. I haven't got brave enough for that yet. I really don't want to admit I need them, so I'm fighting it. If I have contacts on, I have to put on reading glasses. I think that looks a lot older than if I just take my glasses off to read. That doesn't look old, right? No, not yet. The optometrist also said that what they will do for people that are getting older and need the reading glasses, they will give you contacts of 2 different prescriptions. And they say that your brain adjusts. I'm like, you don't know my brain. My brain would probably be like walking sideways or something. But you're all going to experience, if you have good vision right now, as you're getting into your 40s and your 50s, probably that's when your arms are going to need to get longer and longer, because that's going to be the hyperopia that comes with old age. Yes, ma'am.
You can get myopia too, though.
Yes. Yeah. I mean, it's the shape of your eye. It's the change of the shape that affects the focus on the back of the retina. So if myopia, the image is focused on the front. It doesn't get to the retina. The image is focused too far back. So you have to have corrective lenses to get that focus back on the retina again. All right.
And it just there's not it just happened.
No. Mm-mm. And then you can really be messed up, and you can have an astigmatism. Yeah, I've got that too. Because, I mean, the lenses are, your prescriptions aren't always the same in each eye. And mostly, if you wear contacts, they're going to be different. And it's probably going to be partly an astigmatism. And this is when you have a different curvature. It's not the shape of the eyeball like we have here. This is the curvature of the lens or the cornea. And that's causing the image to focus in a different plane as well. But we got corrective lenses that can pretty much handle all that. Or if you got enough money, what can you do? LASIK, laser surgery. But let me tell you this, if you get laser surgery, like, in your 20s, in your 30s, you're probably going to still deal with that old age stuff, and you're probably still going to end up with glasses or going back and having it again. I haven't had it yet, primarily because I've got way too many kids and no money, so, yeah. So this is just an illustration. Myopia nearsighted. See, this is showing where the focal point would be, and see how the retina is too far back? So we're changing the shape of the eyeball. Here, farsighted, the back of the eyeball is too close, and so the image is focused back here behind it. That's not going to work, so you need these different lenses for that. And then astigmatism, the eyeball is the normal shape like we see here, but the lens, the cornea, something's not right, so you're not focusing adequately on the retina. It's more fuzzy and out of focus. Then we need a complicated shape to our lens to get that focus just right.